Hello, my name is Avanish Falunky. I'm the CEO and co-founder of Rain Therapeutics, along with Dr. Robert Doble from the University of Colorado at Denver, or Bob Doble. And it's my pleasure today to be able to present Rain Therapeutics. Rain Therapeutics is based on a first molecule, our lead therapeutic, called tarloxotinib, or Tarlox, as we call it for short. And Tarlox is a hypoxia-activated prodrug. Upon regions of very low oxygen concentrations, this prodrug fragments, and one of the fragments is a very potent pan-EGFR HER inhibitor. It is very potent against EGFR, HER2, and HER4. And we are redirecting this molecule towards a patient population that is very difficult to treat, called the exon 20 insertion mutation population in non-small cell lung cancer. About approximately 6,500 or 7,000 patients every year in the United States whom currently have no available therapy. This mechanism, this hypoxia-activated approach, is a new strategy aimed at solving one of the big problems with treating these patients, which is dose-limiting toxicities. These exon 20 insertion mutations in EGFR and other HER receptors leads to a profile that's very similar to wild-type EGFR and HER. Therefore, by having such a similar profile, it becomes very difficult to distinguish between the receptors in the tumor versus the receptors in normal tissue, such as in the digestive tract or the skin. And therefore, these dose-limiting toxicities often occur in patients with EGFR wild-type cancers or other wild-type-like cancers, of which these exon 20 insertion mutations are a part. This molecule was initially developed by the University of Auckland in New Zealand and was licensed to a company called Threshold Pharmaceuticals here in the San Francisco Bay Area. Threshold had run 2 phase 2 prior studies, and they terminated the drug due to a lack of efficacy. However, we believe they enrolled the wrong patient population. We believe they enrolled patients that did not have EGFR dependent cancers. Rain Therapeutics was founded in April 2017, and in May 2018, earlier this year, we completed a Series A financing of $18.4 million in total, led by Biotech Value Fund in San Francisco and Perceptive Advisors in New York City. We are led by a very strong team of scientific experts, including Dr. Robert Doble from the University of Colorado, as well as the inventors of tarloxotinib, Adam Patterson and Jeff Smale. We're also comprised of a board of directors that includes prominent leaders from the biotechnology industry, such as Mr. Tron Nguyen and Mr. Peter Radovich. We have a clinical advisory panel that includes formidable clinicians that have gotten other drugs approved. Dr. Karsten Witt previously wrote the pivotal phase 3 protocol for OSI Pharmaceuticals in the approval of Tarceva, or erlotinib, which is now one of the standard conventional therapies for EGFR mutant positive non-small cell lung cancer. We also include Dr. Peter Cowie, who was a former member of the Cardiovascular and Renal Drugs Advisory Committee from the FDA here in the US. One of the initial studies that got us very excited about this drug is the opportunity, is the efficacy that we had seen in a proprietary patient-derived xenograft model in exon 20 insertion mutations. This specific model demonstrated that at clinically relevant doses, tarloxotinib led to very rapid and dramatic tumor shrinkage in a tumor model that was derived from a patient with exon 20 insertion mutations. Notably, because tarloxotinib depends on hypoxia activation, we do not anticipate there to be significant fragmentation and activation in healthy tissue where there is no hypoxia. And therefore, we believe Tarlox leads to a far better tolerability profile than conventional agents. This slide shows that relative to other competing agents in the exon 20 drug space, Tarlox has far more manageable diarrhea, rash, and itching incidence because of the lack of activation in healthy tissue. One of the issues with the prior studies that was run by Threshold Pharmaceuticals is that we believe their prior phase 2 lung cancer trial enrolled the wrong patients. Threshold pursued patients that had developed acquired resistance after being treated with Tarceva or another second generation EGFR inhibitor. However, they subsequently removed from the trial all patients with a known EGFR-driven mutation, such as they removed the T790M patients with mutations, and they also removed patients with other EGFR mutations, such as the exon 20 insertion mutations. Those patients who remained were patients that predominantly did not have an EGFR-dependent mechanism, and therefore treating them with an EGFR inhibitor was not a rational approach. However, despite what we believe is an inappropriate patient population, this drug was very well tolerated and led to stable disease in various patients, presumably because of some wild-type EGFR component to their tumor. Over 40% of patients had appropriate disease control despite having progressive disease upon entry. The most prominent adverse event that was noted was grade 3 QT prolongation. However, it was extremely transient. In the bottom chart, you will note that the QT prolongation became immediately apparent after the 1 hour IV infusion. However, it very quickly began to resolve, and by 24 hours, it went largely back to normal, and patients would not receive more drug for another 6 days. And therefore, this QT prolongation did not appear to present much risk to the patients. You will note that in the commentary provided by the prior investigators from a variety of sites, they did not view Tarlox to have a relevant QT profile. Rain Therapeutics will commence a phase 2 clinical trial in 40 patients with EGFR and other HER exon 20 insertion mutations at the same dose that was used before at 150 milligrams per meter squared with a 1 hour IV infusion. We will pursue 5 clinical trial sites in the US, and we are currently evaluating 2 additional sites in Asia. Therefore, we believe this molecule is the only novel strategy to address exon 20 insertion mutations in non-small cell lung cancer. We already know the safety profile. We already understand what the dosing profile is. And now with this latest data from Dr. Bob Doble, we believe we understand the patient population that will be most sensitive to the drug. Our recent Series A financing allowed us to raise the capital to start a new phase 2 trial in the first half of 2019, early next year. And we will be able to provide a look of the first 10 to 15 patients worth of data by a year later, by early 2020. And we hope to be able to identify some meaningful efficacy in those patients. We believe any response rate above 40% could dramatically enhance value for this asset, not just in exon 20 insertion mutations, but also validate the opportunity in patients with wild-type EGFR-driven cancers. With that, I thank you for your time, and I look forward to interacting with you and answering any additional questions in the future about Rain Therapeutics. Thank you very much.